Hello students, the chapter on protein structures is basically designed to impart knowledge on the chemical structures of proteins. Protein structure is directly related to the function in living beings, and hence a knowledge of structures is most important for understanding the function. Proteins basically serve various functions in the body, and the structure is most important for determining its function. For example, collagen, which is a muscle protein, is a supercoiled helical structure. It is long, stringy, strong and resembles a rope. This structure is very conducive for providing physical support to the muscles. And hemoglobin, on the other hand, which is also a protein, is a globular protein that is folded and compact, and its spherical shape and compact shape is very useful for maneuvering it through the blood vessels. Enzymes, which are also proteins, have binding pockets that favor binding of substrates. Enzymes, as we know, are important for metabolic functions and for driving metabolic pathways. Amino acids are grouped according to their chemical nature and are basically the building blocks of proteins. This chemical nature, that is the sequence, composition, type and number of amino acids that make up a protein, determine the physical and chemical properties and the alignment within a protein, which is in turn responsible for the protein's basic function. Now, proteins are an important biological class of macromolecules which are present in all biological organisms, all living organisms, and are made up of elements like carbon, hydrogen, oxygen, nitrogen and sulfur. The uniqueness of proteins is the presence of nitrogen in its structure, which is not present in either carbohydrates or lipids, which are the other major biochemical macromolecules. So, proteins uniquely contain nitrogen, which is giving their special function, and the basic building blocks of proteins, amino acids, are so named because of the amino group which contains nitrogen. Our body requires proteins for both growth and maintenance, and hence proteins are required during all phases of growth, like young children, pregnant women who carry the developing fetus within them, lactating women who are producing milk to feed the child, recovering patients from surgeries, accidents and other trauma, in order to provide for tissue and organ development as well as repair of lost or damaged tissues. There is a constant breakdown of proteins in the body, and this explains the reason why we need to consume proteins on a regular daily basis for maintenance. It therefore becomes of prime importance to ensure that the daily requirement of protein is met from the diet, which provides proteins through a number of sources which are both vegetable and animal origin. So, if we look at the composition and location of proteins, the human body is composed of many different types of proteins located in different parts of the body. For example, the muscle contains about 1/3 of the body's protein, the bone contains about 1/5 and the skin contains about 1/10 of the total protein. The rest of the proteins are located in other body tissues and fluids, basically the intracellular fluid and the extracellular fluid. Even blood contains a considerable amount of protein, which is called as albumin and globulin. The hemoglobin is a combination of pigment called heme and a protein called globin. Every cell in the human body contains proteins. It is a major part of the skin, muscles, organs and glands. Proteins are also found in all body fluids except bile and urine. The presence of protein in bile or urine indicates a diseased state. Now, if you look at the classification of proteins, proteins are classified basically based on the structures. The first type of proteins are globular proteins. These are spherical, water soluble, they have a hydrophobic interior. By hydrophobic we mean the amino acids which repel water, and hence they are comprised of the interior part of the protein. The exterior amino acids are hydrophilic, which helps the protein to be soluble in water or aqueous solutions like the intracellular and extracellular fluids. And these globular proteins mainly have functional roles in the cell like enzymes, hemoglobin and peptide hormones. The second type of proteins are basically the structural proteins, which are called fibrous proteins. They form into threads or cables with repeating units. Essentially, they resemble electrical cables which are bunched together with an external casing. They are water insoluble molecules that provide mechanical or structural support. Example, the keratin of hair and nails and the collagen of muscles. Then the third type of proteins are membrane proteins. Every cell has a membrane. This is called the cell membrane. And these cell membrane proteins often serve as receptors. They are located at the peripheral surface of the cell membrane, or they can be transmembrane proteins where they traverse the entire membrane from the inner cytoplasm, crossing the cell membrane and going to the external environment of the cell. The receptors serve to bind ligands and send messengers to the nucleus in order to produce new proteins. Certain enzymes of metabolic pathways are also located in the membrane, and these are enzymes of the electron transport chain as well as the enzymes of the photosynthetic pathway. They are also called transmembrane proteins. Now, this structure shows the globular protein, which is spherical in nature and which can dissolve in water. The amino acid glycine is located beside the globular protein. To give an example of how big the globular protein is in comparison to an individual amino acids. Around 250 to 350 amino acids comprise a single globular protein. Now, this structure depicts the fibrous proteins which look like electrical cables. This is the casing inside which several fibers of protein are arranged in helical structures and coiled ropes parallelly to form a bundle. These are the membrane proteins, and you have the peripheral proteins as well as the transmembrane proteins. The peripheral proteins are called receptors, and the transmembrane proteins can be enzymes or ion channels which allow ions, glucose and other molecules to pass in and out of the cell. Protein structural organization in any living being is organized into 4 levels. The first level is the primary structure. Now, this primary structure is like a chain of beads, each bead representing an amino acid. It is horizontal and it contains a number of amino acids arranged in different sequences, each sequence unique to a particular protein. When this chain organizes itself into a secondary level like an alpha helical structure or a beta pleated sheet by folding and twisting and held together by hydrogen bonds, it is called the secondary structure. The secondary structure is highly regular. It is a substructure consisting of either alpha helix and strands of beta pleated sheets, and these can be many different types. A number of alpha helices, a number of beta pleated sheets joined together by loops of peptide chains can comprise a secondary structure. A motif refers to a small specific combination of secondary structure elements such as a helix which turns followed by another helix. Now, the tertiary structure is the coiling 3-dimensional structure of a single protein molecule or a single subunit, where the secondary structure folds into a special 3-dimensional arrangement, and this arrangement is held together by various bonds and forces like non-covalent bonds, van der Waals forces, covalent bonds, ionic bonds, metallic bonds, dipole-dipole bonds and other electrostatic interactions. It describes the complete folded and compacted polypeptide chain. The quaternary structure is individual units of tertiary structure joined together by what are called as disulfide bonds. A disulfide bond is formed by reduction of an SH-SH comprised from cysteine. Now, if the 2 hydrogens are removed, then it becomes S-S which is a disulfide bond holding two subunits of a tertiary structure together. A quaternary structure can have one subunit or 12 subunits depending upon the type of protein. So, a number of subunits together form the quaternary structure. An example of a protein which has 12 subunits is the enzyme aspartate transcarbamylase, which is involved in the biosynthesis of purines, which are bases of nucleotides. Disruption or reduction of the disulfide bonds to form SH-SH results in denaturation and loss of function. This structure represents the primary structure, which looks like a chain of beads. The secondary structure is a left-handed alpha helix or a beta pleated sheet, which look like the pleats of a sari. The tertiary structure is the folding of the secondary structure held together by weak non-covalent forces or bonds. The quaternary structure has two or more subunits of tertiary structure, which are held together by disulfide bonds. We have more pictures here to show you clearly how these structures are formed. This is an example of the beta conformation or the beta pleated sheet. They can be parallel or anti-parallel. Now, you have the peptide bonds. When I spoke about the primary structure, we have seen that amino acids are linked together like beads of a chain. Now, what holds these amino acids together? This is called the peptide bond. A peptide bond is the bond between two amino acids, which links them together in the chain of a polypeptide. It is the primary linkage of all protein structures, the chemical bond between the carboxyl group of one amino acid and the amino group of the next amino acid. And this peptide bond is a chemical bond formed between two molecules when a water molecule is removed. Now, the peptide bond holds together the amino acids into a long chain, and this chain has to be folded into the secondary, tertiary or quaternary structure, as we have already seen. Now, this folding and stability is contributed by a number of forces, and it is stabilized by non-covalent forces, including the hydrophobic effect, hydrogen bonding, van der Waals interactions, charge-charge interactions. Protein folding and stability can be seen here in this structure, which shows how once the protein is folded, certain bonds hold the protein apart or together to maintain the integrity of the folded structure. The distance between the folds are called angstrom distances. The hydrophobic effect is created when the functional groups of different amino acids are located in the interior of the protein away from water. Non-polar side chains then interact with each other, making the structure very compact. Polar side chains remain in contact with water on the outside of the protein surface. Now, we have spoken about bonds. These bonds are individually weak, but collectively they are strong. The weakness gives the protein flexibility to change conformations based on the need of the protein within a cell. Once in place, the collective effect keeps the protein in its proper shape and helps to retain its functions. It appears that the hydrophobic effects are very important initially, such that the protein collapses onto itself. Once the bonds are broken, the protein loses its conformation and it is degraded or denatured. Now, when we speak of hydrogen bonding, hydrogen bonds form first as a protein folds itself into the secondary structures, defining the regions of the secondary structure. Many hydrogen bonds ultimately form between polypeptide backbone and water, between the backbone and the R groups, and between the R groups and water. So, these hydrogen bonds within the interior of the protein are more stable than those on the surface, because the bonds do not then compete with water molecules. When we speak of van der Waals interactions and charge-charge interactions, we talk about the forces that establish contact between non-polar side chains, which are very important in holding the structure together. Charge-charge interactions contribute minimally to protein stability because most ionic bonds are found on the surface of the protein and not on the interior. Now, when we look at the polypeptide chain, this chain is formed by amino acid condensation reactions which create the peptide bonds between the amino acids. Peptide bonds are formed by condensation reactions creating bonds between amino acids, and the peptides can differ in size based on the number of amino acids that are linked together. For example, dipeptides have 2 amino acids, tripeptides have 3 amino acids, tetrapeptides have 4 amino acids, oligopeptides have 4 to 9 amino acids bonded together, and any peptide that has more than 10 amino acids linked together is called a polypeptide. Now, look at the structure of the polypeptide chain. You can see that the chain has on the left side an amino terminus NH2 and on the right side a carboxy terminus COOH. These are the two groups that are not involved in the linkage and are therefore intact. But when you look at the peptide bond, which is the CONH bond, the OH group of the COOH and one H from NH2 are removed in the form of a H2O or a water molecule. This is called the condensation reaction which forms the peptide bonds. Now, this is the picture of 2 amino acids in which the water molecule has not yet been removed. This is the ball and stick model of the polypeptide chain, which indicates the different amino acids shaded in blue linked together by peptide bonds. Now, we have some molecules called chaperones. These chaperones can be DNA or any other biochemical macromolecule which binds to a protein and helps it in the folding process. They prevent the formation of incorrectly folded intermediates that may trap a polypeptide into an improper form, thereby losing its function. They also bind to protein subunits to prevent them from aggregating and precipitating out of the solution. Now, this is a picture of DNA chaperones which bind to an unfolded protein and prevent it from improper folding. Now, when we look at the amino acids, there are 20 commonly occurring amino acids in nature. All have an amino group which is NH2 or a charged amino group which is NH3 plus, which is called the basic group. They also have a carboxyl group which is COOH or COO minus in the ionic form. This is the acidic group. Now, these two are attached to a central carbon. The other two valences of carbon are attached to a hydrogen and a functional group which is also called the R group on the 4th bond. This functional group can contain a number of molecules which differ from amino acid to amino acid, and the carbon is called the chiral or asymmetric carbon because its 4 valences are linked to 4 different groups. The exception is glycine, which is not asymmetric because glycine has only a H as an R group, and since 2 valences hold a H group, it is not an asymmetric carbon atom. Now, this is the basic structure of an amino acid with the amino group or the NH2 group, the carboxyl group or the COOH group, the H on one valence and the R group or the side chain on the other valence. Now, we speak about essential amino acids. Out of these 20 amino acids, 8 of them are essential to human beings. This does not mean that the other 12 are not essential. We call them essential amino acids because they have to be provided through the diet, and once they enter the body, they can synthesize all the other 12 non-essential amino acids. But proteins that are being supplied from the diet contain both essential as well as non-essential amino acids. A food containing a protein may be of poor biological value or poor quality if it is deficient in one or more of the 8 essential amino acids. The essential amino acids can be listed as lysine, tryptophan, methionine, leucine, isoleucine, phenylalanine, valine and threonine. In addition, arginine is essential for infants and histidine is considered essential for animals. So, for adult human beings, 8 essential amino acids exist, for infants 9 and for animals 10 essential amino acids are required. Proteins of animal origin have the highest biological value or highest quality proteins because they contain a greater amount of essential amino acids. Foods with the best quality proteins are listed in diminishing order. For example, whole eggs have the best quality or the highest biological value, followed by milk, soybeans, meat, vegetables and food grains like cereals and pulses. Proteins have various physicochemical properties which are again essential for their function. An understanding of how proteins behave in solution is critical to the successful technological application of enzymes and to effective modification of their properties by protein engineering. These are two new fields which are important in both the food industry as well as the plant biotechnology industry. Now, there are certain ionization properties of proteins because of the functional groups and the carboxyl and amino group which can exist in the ionic state. Proteins tend to fold such that their charged residues are located at the surface. This means that the proteins display a number of both acidic as well as basic functions on their surface. Each of these individual groups will display ionization behavior characterized by what is called a unique pKa value. The ionization behavior of a protein is a complex of each of these amino acids that are located within the protein. The situation is complicated in proteins because the pKa value of any protein or of any ionizable group is very sensitive to its environment in the protein. There are certain factors that affect the pKa value of amino acids, and these are several environmental factors like formation of salt bridges between opposite charges, that is the positive and negative charge, hydrogen bonding involving ionizable groups, the close proximity of hydrophobic regions to ionizable groups and the degree of solvent accessibility of an ionizable group. There is something else called the pI value. The pI value of a protein is the pH at which there is no net charge, that is no extra positive or negative charge. The number of positive charges is equal to the number of negative charges. This is a very important physicochemical parameter. Generally, proteins are less soluble at their pI and are more easily crystallized. This property is exploited in processes like protein purification by selective precipitation of proteins using ammonium sulphate. Now, the chemical properties of proteins are determined basically by the R groups or the amino acid side chains, which are very reactive, and this is essential for the biological function of enzymes and other proteins. It also means that proteins are sensitive to chemicals that react with these functional groups or R groups. Proteins exist in their natural environment in solution in some form or the other. Most proteins exist in aqueous solutions and they vary enormously in their solubilities. Some small globular proteins are very soluble, while many structural proteins like fibrous proteins are essentially insoluble. The solubility of a protein greatly increases as the pH moves away from the isoelectric point. At such pH values, there is greater net charge on the protein, resulting in repulsion between protein molecules, keeping them in solution. If these molecules come together and aggregate, they tend to precipitate. So, to keep them in solution, the charge on the protein should exert a repulsive force. The solubility of proteins is affected by the presence of co-solvents like salts. At low ionic strengths, the charge on the surface of a protein attracts counter ions, decreasing the electrostatic free energy and thereby increasing the solubility. Addition of low concentration of salt therefore increases the solubility, while high salt concentrations decreases the solubility of proteins. This is called the salting in and salting out of proteins. Salts vary in the ability to salt out or salt in proteins. Different salts have different capacities. Let us now summarize what we have learnt in today's class. As I already mentioned, protein structure is directly related to its function. Now, proteins are classified based on the way they are organized structurally into globular, fibrous and membrane proteins. Different types of proteins can be categorized into these three different types of classification. The level of organization of proteins in living systems also differs. We have the primary structure, which is a chain of amino acids, also called a polypeptide. We have the secondary structure, which is the folding of the primary structure into a helix or a beta pleated sheet. The tertiary structure, which is the folded form of the secondary structure, and the quaternary structure, which are different subunits of tertiary structure linked together. Now, these structures will determine the chemical nature or the active form of the protein. A disruption or change in conformation of the protein or the structure results in what is called as denaturation. A polypeptide is formed by the joining of two amino acids through what are called as peptide bonds. Peptide bonds are formed by condensation reactions between two amino acids by removal of a water molecule. Proteins are folded with the help of various weak non-covalent forces like van der Waals forces, ionic bonds, charge-charge interactions and hydrogen bonds. The chemical and physical nature of proteins determines its structure, and this nature of proteins is determined by the R groups or the functional groups which have side chains with ionic properties. So, therefore, the structure, the amino acid composition, the amino acid sequence and the types of amino acids and number of amino acids that are linked together, the type of folding of the protein determines its function. I hope this is clear.